grabando, mi querida doctora. Hola, ¿qué tal? Buenos días a todos. Gracias por acompañarnos. Estas son nuestras conferencias sobre discapacidad intelectual y cognición, organizadas por el Laboratorio de Psicolingüística de la Facultad de Psicología. Como siempre, saben que antes de iniciar con la presentación de nuestros ponentes, les recordamos las medidas de seguridad, que son básicamente que no estén ni parados ni sentados en los pasillos y que ubiquen la salida de emergencia. Pero bueno, esperemos no necesitar esas medidas. Con nosotros tenemos el día de hoy un podio muy concurrido y estamos muy contentos de contar con nuestros invitados. En primer lugar, quiero presentar a la doctora Natalia Arias Trejo, que es la jefa de la División de Posgrado e Investigación, que nos va a dirigir unas palabras de bienvenida. Y a continuación presentaremos a nuestros demás ponentes, que son el doctor Mario Arias García, el maestro Carlos Serrano Juárez y la doctora Jamie Edgin. Y les agradecemos mucho su presencia con nosotros el día de hoy. Doctora Natalia.
Buenos días. Buenos días. Creo que está prendido o no. Sí, no, con esto. Brevemente, bienvenidos. Qué bueno que están aquí, qué bueno que nos acompañen. Esperamos aprender un poco el día de hoy sobre diferentes temas relacionados con la incapacidad intelectual, enfermedades genéticas. A nombre del doctor Germán Palafox, les doy la bienvenida. Este es un evento financiado por dos partes, por la asociación T21, a la cual algunos de nosotros pertenecemos y participamos, y por la Facultad de Psicología. Y quiero agradecer especialmente a Jamie. Thank you. Porque pues muy rápido logramos hacer con poco aviso. I think it was like 5 weeks or 4 weeks that you said, okay, I will come to Mexico. Y para alguien tan ocupado como ella, pues realmente se agradece esa respuesta tan rápida. Entonces le agradecemos mucho estar aquí. Pues vamos entonces a empezar. Cada ponencia, para no repetir a los tres, tiene una duración de 30 a 40 minutos, lo que ustedes hayan preparado, para después tener preguntas. ¿Okay?
Gracias. Nuestro primer ponente del día de hoy, entonces, es el doctor Mario Arias García. El doctor Arias estudió la licenciatura en Psicología y el doctorado en Ciencias Biomédicas en la UNAM. En 2016 se unió al Broad Institute, perteneciente al MIT y Harvard, como postdoctorante en el Department of Model System and Neurobiology, donde realizó investigación sobre la neurobiología de la sinapsis, circuitos neurales y fenotipos conductuales en modelos de trastornos psiquiátricos en ratones. Actualmente es profesor asociado del Departamento de Psicobiología y Neurociencias de la Facultad de Psicología de la Universidad Nacional Autónoma de México. Forma parte del Sistema Nacional de Investigadores y ha publicado 8 artículos científicos en el área de neurociencias. La ponencia del día de hoy del doctor Arias se titula Identification and Validation of New Potential Therapeutic Targets in Neurodevelopmental Disorders. Como dijo la doctora Natalia, después de su ponencia tendremos tiempo para preguntas por parte de todos ustedes. Doctora.
All right, thank you so much for the invitation. This is a great pleasure for me to be here to talk about the project that I've been working on for the last 2 years. This project was developed in the institute where I was doing my postdoctoral training at the Broad Institute of Harvard and MIT. And now I am joining to the Faculty of Psychology, and it is really my pleasure to share with you all of the work that I've been working. I'll be happy to take any questions whenever you have whatever comment. So I will start with the title of my talk. It is Identification and Validation of New Potential Therapeutic Targets in Neurodevelopmental Disorders. And first of all, I would like to start talking about the thalamocortical circuit, which is conformed by the cortex, the thalamus, and the thalamic reticular nuclei, which sits in the middle, the thalamic reticular nuclei. I will refer to this structure as a TRN for now. So these TRN neurons receive glutamatergic inputs from the cortex as well as from the thalamus, while the TRN neurons sends GABAergic projections towards the thalamus. Here we have a coronal section of a mouse brain where we can identify where this nucleus is located. And ¿podrían bajar la iluminación de acá para ver mejor las imágenes? Gracias. So this group of neurons are part of are GABAergic neurons that actually are surrounding the thalamic nuclei. And these neurons are the main source of inhibition of the thalamus. These neurons are topographically organized by distinct sensory modalities. For example, we can find neurons that are specialized in the visual processing, auditory processing, or somatosensory processing. And very important is that these neurons are also parvalbumin positive, which is a protein that helps us a lot to identify this structure. This is the shape of these neurons. And electrophysiologically, these neurons have this beautiful electrophysiological property, which is the rebound bursting activity. And there are three main players to generate this rebound bursting oscillation, which is the T-type calcium channels, or mediated by the CAB3.3. These T-type calcium channels are known to be low threshold, so it is very easy to activate these channels and evoke an influx of calcium inside of the cell. I'm sorry, I forgot to mention that this is an intracellular recording of a single cell, so we can manipulate the excitability and to study the biophysical properties of these cells. So there are another two main players, which is the sodium channels, which are in charge of the generation of this charge of action potentials. And we have another key player, which is the SK channels, which are calcium-dependent potassium channels. These are very important because allows the repolarization of the membrane potential. And when we have this repolarization, the T-type calcium channels can be activated again, and then we can have this oscillatory behavior of these neurons. And with this oscillatory behavior, then we can, as I said before, these neurons are highly connected with the thalamic neurons. So we can engage the whole thalamocortical circuit in certain types of brain oscillations. But it is more associated with the delta waves or the generation of the sleep spindles. Therefore, this structure is implicated in the sleep architecture. So as I was telling you before, when we have all of these thalamocortical loop connected and interacting each other, then we can have these brain oscillations. We can grab the signal by using EEG, and we can record from the EEG different brain rhythms that are related with the function of this circuit. So I hope that you are believing me that this circuit is very important for the sleep architecture, the sleep generation of the sleep spindles, sensory processing, and attentional process. But I would like to explain you a little bit more about the sensory processing. And for example, in this beautiful study where Dr. Halasa trained mouse to attend either to a visual stimuli or an auditory stimuli, And at the same time, they located several multi-electrodes right there in the TRN, so they could record the activity of the TRN neurons while this mouse was performing this behavior. So they were recording, if you remember, I told you that the thalamic reticular nucleus can be sectorized. So they were recording right there where the visual TRN neurons are. So all of these neurons were TRN visual neurons. So when the mouse was attending to the visual stimuli, you can see here, in between of these two lines is where the mouse was attending to the visual stimuli. So the TRN neurons stop or reduce the firing frequency, allowing the thalamus process the visual inputs. But I would like to attract your attention now to this part of the panel. And when the mouse was attending to the auditory stimuli, then the TRN visual neurons increased the firing rate. So which means that it was inhibiting the visual part of the TRN, allowing the auditory processing being part of the sensory processing. So with this property of these TRN neurons is very important because it is reflected not only the sensory processing, but also it is making a contrast between the different sensory stimuli that are in the media to being able to focus the attention in one certain stimuli or the other one. So unfortunately, in the thalamic reticular neurons are expressed several convergent schizophrenia and ASD risk genes. For example, the top examples are genes that are related with schizophrenia, which is the CAG9-1I, which encodes the D-type calcium channel, or the GRIM3, which encodes the metabotropic glutamate receptor 3. We have these two examples, which is the CHD2 and the PACHD1, which are also expressed in the TRN, but in this case, these two genes are related with autism. Today, I will focus my talk in the PACHD1 gene, and I would like to tell you that this gene, interestingly, is highly expressed within the TRN. And actually, when the mouse born at the very beginning, the P0, the expression of this gene is almost restricted to the thalamic reticular nucleus. As the development goes, then this gene is also expressed in some other regions of the brain and remains until the adulthood, including the cerebral cortex, hippocampus, cerebellum, striatum, etc. So this PACHD1 gene is an X chromosome gene that is predicted to encode a 12-pass transmembrane protein. However, little is known regarding the specific function of the respective encoded proteins. Actually, in 2008 was the first time that this gene was associated with as a gene risk of ASD. Patients that have the deletion in this gene show a variety of symptoms such as sleep problems, ADHD, aggression, ASD, hypotonia, and intellectual disability, and motor problems. So in order to face this challenge and learn more about what is the functional role of this gene, Dr. Wapping-Feng at Broad and MIT, they developed a mutant mouse that doesn't express this gene anymore. So with this tool, we can then study what is the functional role and what are the behaviors that are implicated with the function of this gene. And when they did the characterization, because I've been telling you that the TRN is very important for the attention selection, they performed this behavior to evaluate whether the attention was reduced in this mouse model. And they found, interestingly, that the mutant mouse, as you can see here in red, have more errors when some distractors in a similar behavioral paradigm that I explained before was present. So these mouse have attentional deficit. But also when they measure by using the open field test, which is basically the travel distance in the time, they also observed that the wild type, the mutant mouse, were more active, which is kind of hyperactive profile. So then this model was very nice to study the ADHD. This model also presents hyperlocomotion, motor deficit, learning impairment, and it was very obvious that this mouse was super aggressive. It was very difficult to work with them. So then, in order to study the electrophysiological properties of the TRN neurons in this mouse, they performed patch clamp recordings to evaluate the characteristic rebound bursting of these neurons. Here we have an example of the rebound bursting of the wild-type neurons, and here we have the rebound bursting of mutant neurons. And as you can see here, the rebound bursting, the rebound oscillation is reduced. Therefore, the thalamus doesn't have the proper inhibition to make all of this sensory processing and attentional process in a proper way. But not only that, they also revealed that the sleep spindles, which is part of the non-REM sleep, it was reduced. And the sleep bout, which is basically the time that the mouse are sleeping, was reduced as well. So it is showing that this mouse has sleep fragmentation. So it is known that the current pharmacology to treat ADHD is basically based on the use of amphetamines. So taking advantage of that knowledge, we were wondering whether we could fix or rescue any of the deficit that we observe in this mouse model. And guess what? When we evaluate the hyperactivity in presence of the intraperitoneal injection of amphetamine, then we observed that the phenotype was even worse, was more hyperactive. So this mouse model was resilient, was resistant to the current pharmacological treatment. And actually, more or less 30% of the patients with ADHD are also resistant to the current pharmacological possibilities. So this was opening a challenging for us to find any potential new therapeutic targets to treat these deficits. And we look at the different pharmacology that has been used to treat ASD, because this gene is also related with ASD. And as you can see here, basically we have antipsychotics, psychostimulants, antidepressants, or non-prescription medications such as melatonin, but it's mainly used to treat the sleep problems. So there is not known any pharmacological tool to treat the ASD. So with that on the table, we were very curious about if we could find any potential new therapeutic target, and if we could develop any pharmacological compound to modulate this circuit and restore the function. In order to do that, we started taking advantage of the PBTD tomato mouse, which basically express the red fluorescence in all of the cells that are parvalbumin positive. And because the TRN is parvalbumin positive, then we could label the TRN in red. And taking advantage of this, we perform microdissections of brain slices, and particularly we extract the TRN, and then we did single-cell RNA-seq to see which kind of genes are expressed mainly in the TRN and not in different areas. And we found that the metabotropic glutamate receptor 3 is highly expressed in the TRN cells. Every single dot that you are seeing here is a cell that was analyzed, and the color is indicating the expression of this gene, the mGluR3. So many of the cells have a good expression of this gene, but the green 2 is not expressed. And we did this analysis because these two guys are part of the group 2 of metabotropic glutamate receptors. They share similar biophysics, similar pharmacological profile, but in this case, the mGluR2 is not expressed, only the mGluR3. To confirm that, we perform a fluorescence in situ hybridization experiment where we have the double staining of parvalbumin, the expression of the GRIM3 gene, and the overlapping. And as you can see here, more than 90% of the cells that were PB positive were also positive for GRIM3. But this is telling us about the genetic expression. But we were wondering if the protein was expressed in the surface of the membrane. So we performed a Western blot analysis, and we revealed that actually these neurons also express this receptor in the surface of the membrane. So after that, we were looking for any commercial pharmacological tool to activate this receptor and see whether we could modulate the circuit. So luckily we found this compound which was synthesized by the company Eli Lilly, which is the LY379268. And this compound has been used not only in vitro but also in vivo assays in multiple species, such as rodents, macaques, and even healthy volunteers. And not only healthy volunteers, also has been used to treat schizophrenia patients as well as generalized anxiety patients. So it was very promising because it's a pharmacological tool that is available and is safe to use. And we also performed some studies to see whether this compound crossed the blood-brain barrier. And it was safe to use, and we could use it as a proof of concept. So the first question that we asked after this was whether we could modulate the rebound bursting activity of these neurons by using this pharmacological compound. Again, we performed patch clamp recordings of single cells, and we confirmed the identity of the recording of the cells by doing a post-analysis staining of PB. And we performed this rebound bursting protocol. And surprisingly, when we add this compound to the bath, we observed an increase of the rebound bursting activity. So this was super promising, and this was the first motivation to establish a collaboration with the medicinal chemistry group of the Broad. And they synthesized another compound, which is, sorry, I forgot to tell you something very important. In this case, this compound is not selective to the mGluR3. It is hitting the mGluR2 as well. So it's an mGluR2, mGluR3 agonist. In the case of the TRN cells, it is not a problem because the mGluR2 is not expressed. So we were confident, and we performed several controls to really be sure that the main modulation of the rebound bursting activity was due to the activation of the mGluR3. But later, with the help of the medicinal chemistry group, we could get this compound, which is an mGluR3 specific positive allosteric modulator. So we performed similar studies to check whether we could indeed modulate the rebound bursting activity. And with the Broad compound, we also could modulate the rebound bursting activity. Here is the summary of the several experiments that we did. And at this point, we were super confident that we could modulate, at least in vitro, the firing of these neurons. And then the question was whether we could rescue the deficit in the PACH1 mutant mouse, right? So here we have the trace of the mutant mouse, and in presence of the compound, in a specific compound, we could rescue the phenotype. We enhanced the rebound bursting activity, not only with the agonist, but also with the Broad compound, specific compound. So after this, we were, well, okay, you could modulate this in vitro, but does that mean that you can rescue any phenotype in vivo in the mouse model? So that was the second challenge. And I think I'm short on time, so I will skip. We also studied the mechanism of the modulation by doing another voltage clamp recordings, and very fast and quickly, basically by activating the mGluR3 receptor with this compound, we enhance the inward currents or the T-type calcium currents and the SK currents, which are two of the main players of this rebound bursting activity. So we know the mechanism, and then we move in vivo. And we started performing EEG recordings to evaluate the sleep pattern. We implanted electrodes in the frontal cortex, parietal cortex, and in the back of the muscles, back of the head, to measure the electromyography. And as you know, when the mouse are sleeping, we have this high amplitude and a slow rhythm, and basically the muscles are without activity. And in contrast, when the mouse are awake, then we have low amplitude and fast activity, fast frequency. And then the activity of the muscles are present. So we evaluate the recordings before and after the injection of the compound, either in wild type and patched one mouse, and we observe an increase on the non-REM percentage of sleep, as you can see here. So in both cases, this compound, by modulating the thalamus reticular nuclei, was enough to engage the circuit in low frequencies and enhance the sleep. Particularly, the band frequency that was increased was the delta rhythm, which is in between 0.5 and 4 hertz, either in the frontal cortex and parietal cortex in both genotypes, in wild type and in patched1 mutant mouse. When we measured the total sleep time, in both cases was increased significantly, and so basically we could rescue this first phenotype of the sleep. So we were super happy for this result, but we were wondering whether we could rescue some other phenotypes, because in this case it was more, we started with this phenotype because it's more related with the TRM function. But when we also did experiments with the broad compound, and we could recapitulate the results. For example, the non-REM sleep was increased, and also the sleep spindles was increased. This is a working in progress. These results are only in the wild-type mouse, but we are showing that only by modulating the mGluR3 specifically, it is enough to get similar results. And then we move to evaluate the hyperlocomotion. So again, we use the open field test. We have wild type, and we have the mutant mouse. And the colors represent different dosages of the intraperine IP injection of the compound. And first of all, I would like you to compare the baseline of the wild type. It is in black, and when you compare the baseline of the mutant, it is very obvious that the hyperlocomotion is there. But then, since the first dosage, which is 1 milligram per kilogram, we observe a reduction of the hyperlocomotion. And if you compare this light color with the baseline of the wild type, it is almost similar. So the locomotion was reduced almost to the basal level. So here is the summary of the total distance that the mouse traveled among the different dosages. But in this case, the take-home message is that the locomotion was also rescued by this pharmacological manipulation. Then we were wondering whether the attentional deficit was also rescued. And in this case, it's a little bit more challenging because there are more brain structures involved. But in any case, it was worth to try. So we spent several months training this imagined ADHD mouse, which is very challenging. But I would like to start going through the behavioral paradigm first. So here we have the mouse were in an experimental cage. I will show you a video so you can have a better idea about this paradigm. But briefly, it was an initiation port, which is this one, where the mouse needed to place their nose there for 400 milliseconds. And in here it was an infrared barrier, so we could know when the mouse was there for the needed time. And right next to the mouse, it was speakers. So we forced the mouse to stay there for 400 milliseconds in order to be sure that the mouse was receiving the auditory stimuli that we trained. Then, depending on the type of sound, it could be 20 kilohertz or 16 or 24 kilohertz. So if it was 20 kilohertz, then the mouse needed to learn that they needed to go to the response port. And if it was 16 or 24 kilohertz, then the mouse has to learn that they need to stay there and not move. If they do this well, then they got a reward in the reward port, which was below the response port. So it was very challenging to train this mouse in this paradigm, but we could get it. So now I would like to show you how does the auditory stimuli sounds. So first I would like to play you will hear the no-go, the go, and the no-go stimuli. But I would like to tell you that once the mouse learns how to do this, then we add another level of complexity, which is the noise. So the same signal was delivered, but in this case in presence of a black noise. And you will hear both examples. But the motivation to add the noise in this auditory stimuli was to challenge the TRN, because the TRN, it is in charge of subtracting the interested sensory input above the noise level. So making this contrast, we wanted to force the system to do this. So this is the first example, is without noise. So that was the example. So as you could hear, it is very challenging. It takes us like maybe 4 to 6 months to get this experiment. But we thought that it was really necessary. And maybe you are familiar with all of this, but these guys needed to train every day, no matter if it was winter outside. So this is an example of the behavioral paradigm. So you can see how the mouse was behaving, and this one is the initiation port. The speakers are right next to the initiation port, so it needed to place the noise there. And if the time was enough, then the auditory stimuli was delivered. Then the mouse needed to decide to go or to wait. In this part is the response port, and below is the reward port. The reward was condensed milk. Yeah. So it is getting the reward. So it was not enough time. This is no go. So he stayed. And then he got the reward, and so on and so forth, right? So when the mouse made mistakes, then they got a timeout. They couldn't continue getting the reward. For example, I think in the next trial, he got that mistake. No, he couldn't get the reward. sometimes they get angry. This is the timeout, even if they stay there. Another mistake, and so on and so forth. So it was like 200 trials per session until they reach this training. So the results of this behavioral paradigm was the following. So here we have the baseline level. In black is the wild type, and in red is the mutant mouse. So baseline means without noise, the pure tone. And the pure tone was similar between both genotypes. But what happened when we add the noise? When we challenge the system, then the performance of the mutant mouse dropped significantly. And then we compare the sessions where we add the noise, but we also inject the compound before the session. And the performance was rescued incredibly. And it was really super nice to see how the behavior performance was changed between one day and the other one. And sometimes when after the injection, the effect lasts for 2 or 3 days after that. So it was super clear, and we were happy to get this result. With all of these proofs, our conclusions are that the cellular and behavioral phenotypes of the PACHI1 mouse model were rescued by the pharmacological activation of mGluR2 and mGluR3 receptors. Either the rebound bursting activity in the TRN neurons was rescued, the sleep problems were rescued, attentional deficit, and the hyperactivity. Therefore, the group of mGluR receptors is a promising therapeutic target for correcting thalamocortical circuit related dysfunction in neurodevelopmental disorders such as ADHD and autism. And finally, in the lab recently, they published this paper where they, by doing gene editing, they could get SHANK3 mutant macaque, which is one of the principal genes that are affected in the autism. So they measured the activity of these guys, comparing control and the SHANK3 by using a motion sensor, like we have in our watches and stuff. So we can compare the activity between the night and the day. And here is a zoom in of the control macaques and the mutant macaques. And as you can see here, they also show the sleep disturbances, the sleep efficiency, sleep fragmentation, and sleep latency. So right now we are synthesizing this compound, the broad compound, to be able to cross the blood-brain barrier of these guys. And we are hoping to rescue this phenotype based on this investigation. Finally, I would like to thank Woping Feng, Sanyan Fu, and Mike Halasa, because they were the principal supervisors of this project. A lot of collaborators, because obviously this kind of work, it was not done only by me. I would like to thank also the therapeutic group, because they were always providing very nice inputs about the pharmacology and all of these related things. Thank you so much for the invitation. I don't know what is the logistic, preguntas, or at the end. Bueno. ¿Y yo ese?
So awesome. It was so awesome in so many different ways in terms of like taking the sleep variables. So I haven't seen very many labs doing neurobiological manipulations to affect sleep. So I'm really excited about all of that. So I guess my question is, how does this mGluR mechanism overlap with what was already done in Fragile X syndrome? Because there was a big clinical trial that went through with that. And can you elaborate a bit about that? And I have some questions about slow wave sleep as well, but I'll save those for later. But I guess I was interested about the Fragile X mechanism and how this is different or similar.
Yeah, thank you for your question. It's very important and very interesting. And actually, there are different receptors. They are part of the same family, but the Fragile X was linked to the mGluR5. And these are mGluR3. So both of them are GI coupled protein receptor linked, and they have different G protein couple. And the expression pattern is different as well. In this case, the mGluR5 is not that expressed in the TRN. That's why we choose the mGluR3, because it's highly expressed there, and it was more easy for us to manipulate the whole thalamocortical circuit by targeting the TRN through the mGluR3. Yeah. You want to ask about, oops.
With the Fragile X group, that's an additional group that might benefit from something. They have sleep difficulties as well, and attention difficulties, so it might be another target for what you're doing. And good models, mouse models of Fragile X. Yeah, it'd be cool.
Because the initial study is like, okay, if there are let's say symptoms that are conserved among different diseases, psychiatric disorders, such as Fragile X, depression, autism, and schizophrenia. In all of them, the sleep is disturbed. So that's why we went to target the circuit, which is in charge of that. And as you said, actually the promise of this is that if we use this mouse model as a way of study this, but the ultimate goal is to translate all of these neurobiological mechanisms to the other diseases and cure at least that phenotype. Yeah.
¿Alguna otra pregunta?
So the other question I had was, how exactly does this drug help facilitate delta? So the spindle link is really clear to me, but the delta wave generation, because those particular waves can be found in culture cell. I mean, you can get delta generation without any thalamic intervention, or so I'm wondering how that happens. And you know, this is not what I study every day. I had this particular neurological link, so I was interested in how that happens. Yeah.
So actually, we have the short answer is that we don't have direct evidence, but we have indirect evidence. One of it is the frequency of the rebound spikes. If we measure these spikes, this is delta rhythm. So in baseline, we have certain amount of delta rhythm. But when we increase this rebound bursting, and this rebound bursting is in the delta rhythm, then the whole network can be engaged in the same brain rhythm, and then we can get the oscillation we can get the delta rhythm increased by doing EEG, the surface. The other indirect measurement that we have done is actually a very cool experiment where we manipulate the TRN cells using CRISPR-Cas9 gene editing, and then we perturb one of the genes, which we know that is very implicated in this brain oscillation, which is the KCFD2, which actually is also linked with autism and epilepsy. And when we disturb this gene, the rebound bursting of the TRN cells was dropped. And then we measure by doing EEG, and the delta rhythm was reduced. So we have correlations It is very complicated to answer, to have this proof, but based on the mechanisms, the current understanding of the neurobiology of the circuit, we can guess that.
There is a question.
Sure.
Okay, they are asking which is the next issue to include in the results of these markers?
Some kind of cognitive intervention among specific disorders, or another kind of measure in this new path? That's the question.
Yeah, for sure. There are a lot of things to do. For example, in different avenues, pharmacologically speaking, there is a new avenue to start doing drug discovery, for example, to refine molecules to target these nuclei in a very safe way. We are very far away from that moment, but I think it's very promising. The other avenue can be, as Dr. Edgin suggests, try to extrapolate this finding or this pharmacological manipulation to other disease models. It can be in vitro, in vivo, mouse models, or as we are doing in macaque models before we could do the step to treat patients. Yeah. But for me, it's very promising that this compound at least has been used in humans. So this is shortening the distance to actually at some point being able to treat patients. Another challenging is that most of the patients with ADHD or autism are infants. So we are talking about several problems with the regulation and stuff. But if we continue doing very careful experiments to avoid any safe issue, I think at some point it can be possible. Yeah. That's enough. Yeah.
Thank you.
I was wondering how valid is or how helpful is to extrapolate these to a control group, but with sleep disturbance problems, because that is also very common.
I think it will be a very good control for sure. Yeah, and actually when I was presenting these results in my group at MIT, my supervisor, Dr. Feng, he was all the time telling me, I think I will use this compound. I should sleep more. But yeah, I think it will be a very good control. And if we only think about the sleep problems, I think it will be very useful to treat this phenotype in different psychiatric disorders for sure, or sleep problems. Yeah. Perfecto. Well, muchas gracias. Muchas gracias. Qué amable. Qué amable, muchas gracias. Al contrario, un placer. Gracias. Quitamos mi computadora. Gracias. Si quieres luego regreso por el mouse y todo.
continuación, presentamos al maestro Carlos Serrano Juárez, quien estudió la licenciatura en Psicología y la maestría en Neuropsicología en nuestra universidad. Su área de interés son las diferencias entre genotipos y fenotipos neuropsicológicos y electrofisiológicos en personas con síndrome de Williams. Actualmente es candidato a doctor en psicología por parte de la UNAM. Asimismo, es neuropsicólogo clínico y coordinador de investigación en ISCALTI, Psicoterapia y Educación, y profesor de asignatura en la FES Iztacala de la UNAM. La ponencia del maestro Serrano lleva por título Differences in Social Cognition Between Williams and Down Syndrome, From Genetics to Neuropsychology. Le cedo la palabra, maestro.
Muchas gracias. So, hello, how are you? It's an honor to me to be here, all faces, friends' faces, to be here with Dr. Arias, Dr. Engin, Dr. Natalia. Thank you for the invitation. So I'm gonna tell you about my doctor project, a little part of them. And this is important to me because one part of this project, I made it here in the Laboratory of Psicolinguística. So thanks for the help. Okay, I'm gonna tell you about genotypes and phenotypes. A genotype is a set of genes that will be characterized a phenotype. And what is a phenotype? Is a physical characteristic that was originated by a genotype. So genes are the hereditary units of the genome and are the functional elements of the DNA that determine a genotype. I like to see something like this. If you have an USB, which you have your archives, you have how to make a doctoral thesis. If you don't have one carpet, your thesis will be incorrect. Something like this is talking about genotypes and phenotypes. In genotypes, we have 46 chromosomes that will derive on a specific phenotype on persons. So some authors have made some models about the relation between genotypes and phenotypes. Also, when we talk about genotypes and cognitive phenotypes, it's not a direct relationship. There are a lot of variables between, like the brain and the cognitive process. Some Covas and Plumbing made a model where they talk about three mechanisms how genes can interact in derive a process cognitive. First mechanism, they say a gene influences one area of the brain, which influences different cognitive process, like this one. They say this is not correct because we found that a gene, like the mechanism 2, influences several areas of the brain, and each area affects a specific process cognitive. We found that the brain, one area is not for a specific cognitive. One brain area is implicated in a lot of process cognitive. So they say that the best way to see how the relationship is the mechanism 3. A gene influences several areas of the brain, and each area have several cognitive processes. It's more approximate to the reality. Another model process is of Johnston and Edward model, where we see how the genetic activity will be implicated on protein synthesis, intracellular biochemistry, and of course on the cell membrane, something like Dr. Arias said. No? And this mechanism will be influenced on different physiological processes, and all obvious will be impact to the behavior. The interaction of all these variables is not linear in the time. It occurs at different moments of the time. So this is important because we can find that genes not only activate in one linear time, could be activated along the lifespan. Finally, Bayes-Snowin model was created for dyslexia disorder. They say that we have the genes that will be activated or will be implicated in several areas of brain. These several areas of brain will be implicated on different cognitive process, and this cognitive process will be implicated on different behavior. But also they say that we have the environmental variables that will be implicated also with the area brains. Okay, this is important to the neurodevelopment. In the last years, Williams syndrome was used like a natural model to make how the relationship between genes and behavior is created. Williams syndrome is a neurodevelopmental disorder and it's a rare disease because the lower prevalence, and they have a specific phenotype cognitive that were described by Bellucci. This phenotype is characterized by intellectual disability, visuospatial alterations, language skills preserved or medium preserved, ADHD, and something interesting, hypersociability. That in a first time was think there were an extra ability, but in the times it's also a disorder. So, some authors have mentioned that the deletion of the chromosome 7, that is the cause of the Williams syndrome, will be implicated in a different cognitive process. We have the losses of 24 to 26 genes from FKBP6 to GTF2I. This is the common deletion of the Williams syndrome. And this part of the genes were implicated on intellectual disability, heart disease, calcium disease, and also visuospatial disorders. These all part of the genes are implicated on disability disease and maybe on autism spectrum disorder that were found in some atypical cases with Williams syndrome. What happened with the Williams syndrome brain? Well, this is mentioned that a review by Jakowski that Williams syndrome brain have alterations or a volume reduced on prefrontal cortex, parietal cortex, amygdala, subcortical nucleus, but they have cerebellum, but they have a major volume on temporal cortex. That this could be implicated on musical abilities that Williams syndrome got. So this way will make me think that maybe we can make a model with Williams syndrome about the relation between genes and behavior. This is adapted for Bishop model, and I mentioned that we have some of the genes that were implicated on the Williams syndrome. The Arias brains were described, they have a more implication. Also, the Arias brain, what process are implicated for them, like executive function, social cognition, visuospatial skills, and learning and memory. And also some behavior like emotion, hypersociability, adaptive behavior, and learning disorders. Also, I put the environmental like a variable, like it's constant implicated on all the model, because, for example, the scholarship have some implication of how this will be developed. A paper from Melanie Porter found a typical deletion of Williams syndrome that preserved these genes, GFTI-RD1, GFTF2I, and GFTF2IRD2. These three genes are considered from the same family. What they found? Well, they found that the patient will preserve these genes, have better skills for social cognition and for visuospatial skills. So they suppose that maybe these three genes will be implicated on alterations of theory of mind and also the visuospatial skills. The last year we published a paper. We compared two groups of Williams syndrome. One group preserved the three genes. One group preserved only this gene, and the other group loses three genes. All include this one. Here we have the two deletions of the two groups. The group A that preserves this one gene, and the second group that loses this gene. What we found? Well, we assessment with a neuropsychological battery, And we found specific alterations for a typical group that will be the group that loses the last gene. We found this group have worse ability to visual spatial skills, specifically on visual closure. And also we found that this group have more behaviors of isolations and have more problems to emotional intelligence. We have one problem when we made that this. We evaluate with an inventory for parents, but we found that these items of this variable is associated with theory of mind and some social cognitions. So we want to know what happened with these variables. When I was making my postulation doctor exam, my committee said, "Oh, wow, that's good. But I think you will try to a mental group age and a chronological age comparison." So I talked with Dr. Natalia Arias, and she let me use a mental age group. And there is another question that I made. Is there any relation between genes and social cognition, between genotype and social phenotype? So one way to compare different developmental disorders is found a specific neuropsychological phenotypes or neuropsychological characteristics of the neurodevelopmental disorders. It's compared with another group with similar mental age. So the major of the papers compare Williams syndrome and Down syndrome. This is because one, they have some similar characteristics of social cognition, like approachability to strangers. Another thing is that they have similar EQ, and of course they have similar mental age. So this comparison can show us what specific cognitive process is characteristic from each one of the disorders. Talking something about the neuropsychological phenotype of Down syndrome, we found they have poor language skills, poor verbal memory, executive function, and also they have intellectual disability. About the social cognition in Down syndrome, were described that they have a good recognition of happy and anger, but they have better the execution for recognition, fear, sadness, and theory of mind. So what we do, what is social cognition? Well, social cognition can be defined as a neurobiological, psychological, and social process through which social events are perceived, recognized, and evaluated, and then generate the most appropriate behavior according to the particular circumstance. It is a psychological process that allows interpreting social signs to respond appropriately to the context. This is important for us, like humans, because it's important to sociality. Okay? Adults made a model of social brain. They say we have the stimulus that will be processed of stimulus factors by three variables: cognition, emotion, motivation, and then we'll have a social behavior to this context. What areas of brain are implicated in this social cognition? Well, sensory and association cortices like fusiform gyrus and superior temporal sulcus, amygdala, orbitofrontal cortex, cingulate cortex, and right superior cortex, and finally motor cortex, basal ganglia, hypothalamus, and brainstem. And also these areas are implicated in this process cognitive that will be affecting the social behavior. These also adults mentioned some areas that will be implicated on the social brain. For example, for theory of mind, we have medial prefrontal and cingulate cortex, left medial prefrontal cortex, left medial prefrontal cortex, and right medial prefrontal cortex, and one paper mentioned amygdala and superior temporal gyrus. Also for recognition emotion, face recognition emotion, we have the amygdala. That the first hypothesis of the sociability of Williams syndromes was that this dysfunction of the amygdala is a problem because they can't recognize the fear. So this is one approximation of the approachability of the Williams syndrome. What happened with the social brain and the Williams syndrome? This is the areas that were implicated on the hypothesis of the social brain of adults. the most of the areas were on the right hemisphere. We have the prefrontal cortex, the parietal right cortex, the amygdalas, that will be the two of them, and finally some areas of the temporal. So this is interesting because the areas that were minor volume in Williams syndrome is the parietal cortex. That will be explained some of the symptoms of the social cognition on the Williams syndrome. Järvinen described a phenotype, social phenotype for patients with Williams syndrome with the most common deletion, 1.5 megabases. This is 24 genes, and they preserve the last gene of the family of the GTF2I. And they mentioned that these patients, this phenotype is characterized by alteration of social judgment, emotional processing, theory of mind, and definition. And these processes are associated with the alterations on the amygdala, on the fusiform gyrus, on orbitofrontal and right parietal cortex in Williams syndrome. So we found that there are some areas specific on Williams syndrome that could explain the social phenotype of the Williams. How to evaluate social cognition? Well, we use a compression test of the Wechsler scales to assess social judgment. We use Happel's strange histories that let us assess theory of mind. And we use Ekman faces to assess face emotion recognition. This is one of the examples of the Happel's strange histories. We use, they have 6 kinds of histories: simulations, joke, lie, white lie, rhetorical phrase, and irony. We read this history and we show the picture to the patients. And this is the example of the simulation history. Maria and Rosa are playing at home. Rosa takes a banana from the fruit bowls and gives it to her. He puts in his ear and tells Maria, "Look, I'm talking on the phone." Why did Rosa say this? Who can say? Nobody? Because it's a simulation of the phone. No? That would be a correct answer, okay? This is a lie kind of history. Anna goes to the store to buy a package of candies. When she leaves the store, see that a girl from her class named Julia is coming. Then Anna hides the candy pack behind the back. When Julia arrives where Anna is, she questions, "Hi Anna, can you give me some candies?" And Anna replies, "Oh no, I haven't anymore. I'm sorry, I have already run out." Why did Anna say this?
You don't want to share.
Yeah, she hated her. This is a white lie history. It's Elena's birthday. Elena looked forward to this day because she had been yearning for his friends and his friends to give him some of her friends sing happy birthday to you and him his gift. Elena was sure there would be a skate inside. But when she opens the gift, see that there are no skates in the box. Oh, but there is a ball. And that was not the gift she wanted. Then his friend asked him, "Did you like our gift?" And Elena replies, "Yes, thanks. The ball is very beautiful. It's just the gift that I wanted." Yeah, something like with supervisor thesis, no? This is a joke history. Today Raul goes to Claudia's house for the first time to snack. Raul wants to meet Claudia's dog, and they have been talking about him all day because Raul likes dogs very much. When Raul arrives at Claudia's house, she goes running to open the door, and the dog jumps on top of him to greet him. The dog of Claudia is huge, almost as big as Raul. So while Raul has the dog on top, he tells Claudia, "Claudia, you don't really have a dog, you have an elephant." Irony history. It is time for lunch. Pedro is eating a snack while watching some of his classmates are playing a soccer game. When his sandwich ends, Pedro approaches his teammates to play with them and tells them, "Hello, can I play with you?" Then one of the children turns and say, "Yes, of course, when the cows fly, you will play. Oh, that's a bad. Finally, rhetorical phrase. In the courtyard, there is a group of boys and girls who are sitting talking about the movie they had seen on Sunday night. Everyone talks and talks except Lola, who is watching quietly how everyone speaks. Then Oscar, one of the children who were talking, look at Lola and say, "Hey Lola, what's up? Have the cat eat your tongue?" So we read two histories of each one of the kind of the happy strange, and also we use two neuropsychology experts to make K-coins. So we found a big relationship between the two neuropsychology experts. For the Ekman phase, we pilot 30 phases previously to present to Williams syndrome and Down syndrome for what phases we're gonna use on our study. So we use 4 emotions: happy, fear, neutral, anger, and sad phases. We put on a laptop and we ask to the patient what kind of emotion is the person. So if they say the correct emotion, they have 1 point. Okay? Okay, then we found to evaluate the genotype, we use a genetic test or genetic technique that is called microarray chromosome arrangement that let us evaluate all the genome of the patient. And this technique, we can find how many genes they lost, and what genes they lost, and what genes they preserve. So we found one person, one atypical case, with a deletion of 1.1 megabases that preserve these 3 genes. Here there is the patient, and the deletion is from KBP6 to CLIP2. This is preserved these 3 genes. We also found for patients who has a major deletion, this is from PKBP6 to GTF2IRD2. This is they lost the 3 genes, and also we have 7 patients that have the common deletion that goes to PKP6 to GTF2I. So these deletions let us compare what happened with these 3 genes on the phenotype social cognition. Also, we use a mental age group that was Down syndrome, and also a typical development group to make a comparison between them. For the statistical analysis, we made 2 statistical analyses, one between groups, and we use no parameter statistical, Kruskal-Wallis, and we use another analysis, a T-student that was adapted for this author to let us compare one unique case with a sample. So we can find a specific significant difference in some of the variables. First, the social judgment this is the result between groups. So we have the group of 1.5 megabases, 1.8 megabases, Down syndrome, and typical development group. We found the difference, but we found that only the typical development group have a better performance with Williams syndrome and with Down syndrome. There's no difference between the two neurodevelopmental disorders. Sorry, this is this was for previous way. We found only difference on mental age and EQ, but what the typical developmental group was better than the 2 groups with the neuromental disorders, but were no difference between the two disorders. This is the results of the emotional recognition. We only show the variables where we find a significant difference. So you don't see, for example, happiness and anger, because the 3 groups were similar. And the study shows that Williams syndrome, Down syndrome have good or adequate performance to identify these 2 emotions. We found that fear were best to typical development with 1.5 megabases and syndrome of the Down. Down syndrome, Spanglish. We found also sadness were best for typical developmental group and Williams syndrome, and we have that Down syndrome have alteration for recognition this emotion, and it's specific for this neurodevelopmental disorder. Finally, the neutral phases were best performance for typical developmental group than the two syndromes. The stories, well, I had to tell you that these three kinds of history is associated with first order theory of mind task. And these three are associated with second order theory of mind task. So this is easier than this one, okay? So we found for the simulation, all two syndromes have similar performance, and typical developers have better performance. But it's interesting that lie and what lies, Williams syndrome have a similar performance than typical development, and these two groups have better performance than Down syndrome and 1.8 megabases. So this is interesting because maybe GTF2 IRD2 is implicating of theory of mind, and this group have more alteration in this variable. About joke, irony, and rhetorical phase, well, we don't have difference between neurodevelopmental disorders, but obviously typical development group have better performance than two ones. So this test of Harper Strongers let us evaluate or assessment, first order task theory of mind and two order theory of mind task. So we simple history that will be simulation, lie, and what lie is here in this variable that it will be first order task of theory of mind, and joke, irony, and rhetorical phrase will be rhetoric histories that will be associated with second order theory of mind task. We found only a difference on post hoc that typical development have better performance than Down syndrome, but typical development and Williams syndrome have similar performance. So we found that Williams syndrome have better performance on first order theory of mind task than Down syndrome. But second order theory of mind task, two of the syndromes have similar performance. What happened with the unique case? This is interesting because unique case have a better performance than the three of the groups. This is 1.5 megabases, 1.8 megabases, and Down syndrome. This shows us that social judgment is similar to typical development persons on social judgment. On phases, we have similar performance between this participant on the two groups of Williams syndrome and Down syndrome. So we found that maybe this gene is not implicated on emotional recognition phases. This gene is not expressed on amygdala, and this test is associated with the amygdala. So this could explain why we don't found differences on emotional recognition between this unique case and the other groups. But we found that this group have better performance on theory of mind task, on simulation, on lie, and white lie, and they have similar performance that typical development group. We found that they have the similar alteration that these two groups on the three stories of two second theory order theory of the mind task. Then we have the comparison between simple histories and rhetoric histories. We found that 1.1 megabases, this is the patients who preserve the three genes, have a better performance of this group, Down syndrome, and is similar to typical development group. And they have a better performance also to these two groups, but also to typical development group. They have better performance or worse performance than typical development group. How can I conclude these found? Well, we found difference between Williams and Down syndrome, first of all. There was no objective maybe of the research, but was very interesting to what we found about the difference between these two neurodevelopmental disorders. First, we found Williams have better skills theory of mind, specifically on first order tasks. They have better recognition for sadness phases, and they have better social skills. Down syndrome, here I put like a question, alteration of theory of mind. One limitation is that the task we use is verbal. So this could affect the performance of the Down syndrome. So it's a question that will be a doctor degree thesis maybe. They have worse face emotion recognition, sadness, fear, and neutral. The sadness was associated with temporal cortex. Temporal cortex is an alteration on Down syndrome, but in Williams syndrome doesn't. So maybe that's what could explain why Williams syndrome have better performance to recognition of the sadness phases. Okay, these authors made a review of how these genes are implicated on the brain and the cognitive process. They mentioned that these three genes belongs to the same family. These genes are implicated on the protein TF2 that were implicated on the migration neuronal phases. Also have a high expression on dorsolateral prefrontal cortex and parietal cortex. These areas are associated with social cognition, like social judgment, theory of mind, and maybe empathy. Empathy like a question because a limitation is we no evaluate, no assess empathy. We can conclude there is a difference between genotypes in Williams syndrome and what the social cognition phenotype. What difference we found? Well, 1.1 megabases who preserves the three genes have better performance on social judgment and theory of mind. 1.5 megabases have better performance on theory of mind, but not in social judgment. And finally, 1.8 megabases have worse performance on theory of mind and social judgment. So this could explain different social phenotypes and of course implicates different intervention on social skills. Finally, I propose a model use a Williams syndrome to explain the relationship between genes and behavior. I have the genotype variable that will be on a circle. In this circle are the genes that were implicated on the deletion of the Williams syndrome. These genes are described that are associated with these areas on the specific arrows. Also, I added an arrows with directional because the brain is not only an area that is implicated in a process. Today, the literature says us that are neural networks that were implicated on process cognitive. So we don't know how these neural networks are made and how explain the process cognitive. It's a question. So, but we know that more specific areas maybe have a more associated with process. Here are the specific processes that are altered on Williams syndromes. That will be executive function, social cognition, visual spatial skills, and memory and learning. And how impact to the behavior on emotion, hypersociability, behavior, adaptive behavior, and learning. So in this specific work, we found these three genes. I put these two genes because they have similar function, but this one have different function. The biography says that this is like a regulates the expression of these two. That's interesting, because this is there is no gene that regulates the activation of these two genes. These two genes are described that are associated with prefrontal dorsolateral cortex and parietal cortex. These areas, the prefrontal dorsolateral cortex, is associated with executive function and also with social cognition. Parietal cortex will be associated with social cognition and visuospatial skills. And also we found that executive function is associated with behavior adaptative, and maybe persociability. Social cognition is associated with persociability, adaptative behavior, and visuospatial skill is associated with adaptative behavior and learning. But also we can't forget the environmental variables. The environmental variables affected all of these models on all the levels. Scholarship, by example, affected the expression of the genes, could affected the stimuli of areas, will affect the process cognitive, and also will affect the behavior. So we can see this model like linear and simple, but it could explain difference between genotypes and neuropsychological phenotypes in neurodevelopmental disorders. So we have some limitations, like studies researchers have sample size. We would like to have a major sample size. Also, now we have a major sample size. This work was made with metropolitan area. Now we have people from Mexico, all the interior of Mexico, and Also, I have the doubt what happened with Down syndrome. Maybe we have phenotype similar like Williams syndrome. Also, we want to use now a visual task of theory of mind. And also we want to use an empathy task to evaluate the 4 variables that Adolf says are implicated in the social cognition. Thanks to the joint Trisomy 21 Research Society, and also to Laboratorio de Psicolinguística for this work, and all this is the organization for this work. Thank you.
Vamos a pasar a las preguntas. ¿Alguien tiene alguna?
Because you have too many results, that's my excuse. I probably didn't follow some of them. So in sadness, where you find different, better performance in Williams than in Down syndrome. Okay, I have a question. Is that in the 2 cases, so in the faces and in the stories that you tell them? Or just the faces? That it maybe do some of the difference that you didn't expect that much because of the verbal implications, and we know that Down syndrome may have that issue. But how does it work in the face? It doesn't. They don't have any verbal.
Well, the instruction of the emotional faces only mentioned 4 emotions. So I only put the face, and they had to say sad, anger.
They had to say.
Yeah, I had to say the emotional and the expression, and on the happy strategy, they had to make a more detailed history or answer about why the children of the story say that answer. So maybe that's why they have better work performance on this task. But the emotional recognition was very easy. It's not a big history, so the memory is not duplicated, although like the happy strange histories, like the emotional recognition. So that's why I mentioned that this will be a specific characteristic of the Down syndrome. But the happy strange history, the stories are more longer. So this could affect the the performance of the Down syndrome.
Yeah, I think the stories are quite intense linguistically, but then also having to generate an answer to those beyond the cognitive part of that, like the answer generation is always very difficult for kids with Down syndrome to be put into that position. They do a lot better with linguistic generation when they sort of are eased into that. So I'm glad that you said you're going to move to a visual task, and I was wondering what you're thinking about for that, because there are some really great eye tracking assessments of theory of mind that might be something you can do.
There was some specific tests that will evaluate visual theory of mind task. I can't remember now but there is a task that is not linguistic, but it's something like the Baron-Cohen test, but with image and with emotion. So these have minor linguistic and more visual task. So in Williams syndrome, we found that linguistic is not a relationship with the performance on the theory of mind. That's interesting. Some authors assessment Williams syndrome could have better linguistic abilities, and Williams syndrome with worse linguistic abilities, and they found that this is not affected the performance on the theory of mind task. But in Down syndrome, the linguistic is worse to Williams syndrome. Williams syndrome, you can talk with them, you can have a better conversation with them, and Down syndrome it's kind of that you can have this kind of this is a difference. So some authors are made some visual tasks that have minor linguistic ability, linguistic variable, and have a more visual, like a motion picture, and this could maybe we can use it for evaluate theory of mind with a visual performance and with a verbal performance.
¿Alguna otra pregunta?
Hi, very nice talk. I would like to learn a little bit more about the brain networks that are associated with the hypersociability that the Williams syndrome exhibit. Can you tell me a little bit more about that?
Yeah. There were three hypotheses. First hypothesis, the amygdala dysfunction will be affected the emotional recognition of the faces, specifically of the fears. If this patient can't recognize the fear, so their approachability, because they don't have this recognition, so their approachability to strange. But in the last year, some studies were found that Williams syndrome approachability is emotion where if their emotion of fear is or is not. So this was the first hypothesis. The second hypothesis, they have a major attention to faces of the persons, but also they found that with different studies that this couldn't explain all the things of the perceptibility. So the social brain, that is a hypothesis of adults, mentioned that were specific areas that will be implicated on a specific process cognitive of the social cognition. They mentioned some domains of the social cognition: empathy, theory of mind, emotional processing, that is the emotional recognition phases, and also, I missed one, a social judgment. These are associated with specific areas. They mention prefrontal cortex, specific orbitofrontal cortex, and dorsolateral cortex, right parietal cortex, because it's associated with empathy and some task with theory of mind, amygdala, and also some areas of the temporal lobe. So it's a circuit that will it's activated when you have with this process cognitive, and this process cognitive is that he calls a social cognition. So in Williams syndrome, they have minor volume on the parietal cortex, especially the right parietal cortex. That's why some authors mention that they have visual spatial skills with alterations, and also maybe in theory of mind. And also the amygdala that were implicated, that it's in a minor volume, and they have an hypoactivation from this structure that could explain the hypersociability of the Williams syndrome. If all this circuit maybe it's altered, could explain why they have this hypersociability, and also the minor volume of the prefrontal cortex will be associated with the disinhibition. So it's a circuit that could explain this hypersociability of this neurodevelopmental disorder.
¿Alguna otra?
So you may want, so intelligence is integrated by different areas. So regardless of the fact that you have the similar mental age, it seems that you have different verbal abilities, no? So you may want to use a test like any, but like the Peabody, to, apart from checking the mental age, checking that the verbal intelligence is similar, and therefore you wouldn't have the confusion.
Yeah, maybe use a test of verbal abilities like Peabody will be. I have when I use the use of the Down syndrome was a methodological use, no. But when I started, I found that I had to have a better control, no. Now I can use now that I found that it's not only the EQ, that they have specific different social cognition on the neurodevelopmental disorder. So yeah, maybe I can use now a verbal test to control this variable.
Well, as you know, this conference is being transmitted in YouTube, and we have here a question from someone. Maybe like this, yes.
We have here a question from Saulo Manuel Santana Hernandez from YouTube, and he said, what do you think about the immersive tools like VRFet to work these features of the term like the happiest French stories? We can use that kind of tool to measure some variables like in your study.
Sorry, again?
What do you think about the immersive tools like VRFet to work these features of them, like in the happiest stories?
The tools? Okay, I don't know what kind of tool, sorry. Virtual reality. Okay. That would be a good tool, but one Happen Strange History have it described that have a very good confiability because are histories that were similar to reality. So that's why I decided to use Happy Strange History, because Borrowed Coin is not like something like reality. So these stories, Happy Stories, are more approximate to stories of the reality. No? Maybe, of course, a virtual reality will help more, but in this case, a good how do you say? Una propiedad, una ventaja, one opportunity is that the approximate to the reality things. But of course, maybe that could help more. But in the case, maybe I was use another. Maybe it will help more a virtual reality. but the HEP story have got this opportunity. That's why a lot of studies began to use it for the approximate to real life stories.
No. No minutes, no rest.
Ay, sí, cierto.
A las 12. A las 12 en puntito estamos de regreso, por favor, para que no se atrase. ¿Sí? Muchas gracias.
con nuestra tercer plática para cerrar con broche de oro estas conferencias. Nuestra próxima ponente es la doctora Jamie Eggen, que es psicóloga especializada en el área de neurociencia cognitiva del desarrollo. Su área de especialización es desarrollo de la memoria y sueño en poblaciones con desarrollo típico y atípico. En los años 2000, trabajó en la caracterización de los perfiles de aprendizaje y memoria en personas con síndrome de Down y autismo. Más tarde, publicó el primer trabajo que relacionó los problemas de sueño con problemas comunicativos en personas con síndrome de Down. La doctora Eggen ha recibido distintos premios, entre ellos el Diane Lynn Anderson Memorial Award por servicio a la comunidad, el University of Arizona's Koffler Prize de divulgación de la ciencia y otros muchos. Actualmente la doctora Eggen es profesora asociada en el programa de grado de Cognition and Neural Systems en el Departamento de Psicología de la Universidad de Arizona. Además tiene afiliaciones en los programas de Family Studies and Human Development, the Franz McClellan Institute, the School of Education, entre muchos otros también. Asimismo, colabora con un gran número de comunidades nacionales, estatales y locales para ayudar a personas con discapacidades de desarrollo. La ponencia que nos va a regalar el día de hoy la doctora es Memory, Intervention and Sleep in Intellectual Disability. Le damos la bienvenida. The floor is all yours.
Thank you. Thank you so much. It's so wonderful to be here. Today is our American Thanksgiving. And so thank you. Happy Thanksgiving to you. So on Thanksgiving, we have a tradition in my house to say what we're grateful for. And I'm really grateful to be in Mexico and to be here with you today, and grateful for everybody who's here to hear this talk and to share this with you, those of you who are listening everywhere, even the people on YouTube. That's pretty exciting. So thank you for inviting me. This is my second trip to Mexico City, and I just love it here. So it's wonderful to be here again. So today I'm going to be talking a bit about our work on memory intervention and sleep, particularly in Down syndrome. We've studied Down syndrome for 20 years or so, and there's a lot of work that needs to be done with the population of individuals with Down syndrome in the area of sleep, in particular how it affects their memory, primarily because they have sleep difficulties across their entire lifespan, starting from infancy into late adulthood, at periods of time in which they are engaged in very challenging memory tasks. And one of those challenging memory tasks is actually learning language. So putting together arbitrary labels with objects and making those associations and learning new words is actually a challenging memory task. And so what we've been trying to understand is how individuals with Down syndrome form new memories, such as making those new words, and how they do it under different conditions in which they have to sleep right after learning, and different encoding conditions. So one of the ways that we do learn new words, like brontosaurus, for instance, and the name of this dinosaur, for instance, is to have that arbitrary label presented by a parent or a caregiver or a teacher, and then make that label associated with that object. And we know from data from the literature with typically developing kids that if children are presented those labels and then they nap right after that presentation, that they actually can retain those labels better, that they're better at retaining those after a nap than if they're left awake after that interval. So there's something in early development that memories are quite fragile and actually need the support of a sleep interval to be retained from longer intervals. And a lot of that work has been done by my colleague Rebecca Gomez at the University of Arizona, which she's shown that infants have these very fragile memories that are supported by sleep and sleep intervals. So that's one way that children learn new words and memories that supports longer term retention. We have also been interested in examining other ways that maybe rest reliant on the neural systems of the hippocampus. So sleep, we think, is particularly reliant on hippocampal processing because during the sleep period, there are processes in which the hippocampus actually replays neural signatures that are encountered during the day, and you can see through different neurophysiological mechanisms that there's a replay process that would help to consolidate new memories or new words in this example. But what we've been really interested in looking at is, are there other ways of rapidly consolidating information or providing a consolidation process that would not rely on this same hippocampal mechanism that we could use of benefit in individuals with Down syndrome who have both sleep problems and problems with hippocampal dependent memory. And so 3 areas that we've been exploring are fast mapping processes, and I'm going to go into the details of that. That's basically presenting new items in relation to ones that are known in a very implicit process. Self-reference, which is another method of consolidating information that is done in reference to the self, using the self concept as a support for memory retention. And then also retrieval practice, which is basically the testing effect in which memories are retained longer based on having an opportunity to retrieve and actively engage in the retrieval process for a memory prior to a sleep interval. So what we've been doing is trying to understand whether or not we can support long-term retention of memories, such as new words in some instances, through different methods that aren't reliant on the processes that are most impaired in Down syndrome. Down syndrome is a disorder of sleep as well as learning. There's a long history of the cognitive profile of Down syndrome, including language deficits, hippocampal deficits, but this group, at least 50% of them have sleep disturbance, including, but not limited to, sleep apnea across the lifespan. So it's been under-recognized in the past how much sleep is impaired in this group. It is a lifespan issue, and it actually could be present at times in which children are really reliant on these neural systems like the hippocampus and sleep processes to hold on to information across the long term. And so we've been very interested in trying to examine that question, highlight what's going on with Down syndrome in terms of sleep and how it relates to learning. Down syndrome has sleep deficits, but it also is a disorder, a developmental memory disorder of the hippocampus. So there are good data to suggest that there's reduced neurogenesis in the subfields of the human fetus of individuals with Down syndrome across subfields. In mouse models, we have excellent mouse models of Down syndrome, ranging from the TS65VDN, which was the first mouse model of Down syndrome, to now more complete mouse models that actually hold the entire human chromosome. And in some of those models, we can see wider place cell fields and less intertrial stability for place fields in those models. Now, place fields are a way that the hippocampus, this memory structure, represents memories and represents the spatial environment, and those seem to be not functioning in the same way in Down syndrome mouse models. There's also hyperactivity of CA3 associative connections, suggesting that those memories aren't as specific and they're not sort of honed in the same way in those animal models. In humans, we have good neurological data to suggest that across the lifespan there are differences in the hippocampus in the individuals with Down syndrome. So there's reduced myelination of the trisynaptic circuit, this hippocampal circuit in Down syndrome in humans. And then there are lots of behavioral data to suggest that memory is impaired in these populations, and impaired in a way that suggests that there's a hippocampal deficit. So that's why we've focused on trying to understand, are there ways that we can consolidate information that might not be relying on the hippocampus, because we have found findings to suggest that there are some difficulties with sleep-dependent consolidation in this group, in addition to their other memory difficulties. Of interest to some of you here might be that they also develop Alzheimer's disease at an increased rate. And so the picture is that they have lifespan difficulties with hippocampal development and the representations and the memories that are formed in the hippocampus, but they're also at risk for later decline, including decline of the functions of the structure. So are there sleep consolidation difficulties in Down syndrome? And we and others have conducted work to try to understand that question, including this, the first study of sleep-dependent learning from Ashworth, Anna Ashworth, and Dagmar Demetrio and Annette Karmeloff-Smith's group, in which they basically trained individuals with Down syndrome on certain word lists and looked at how they retained these word lists across time. So they had a group with Down syndrome as well as Williams syndrome in this study. Williams syndrome also shows sleep impairments, but not to as severe an extent, a little bit different sleep impairments than Down syndrome. And what they found was that individuals with Down syndrome were better at retaining information if they learned the names of certain animals. There was a long list of these different names of animals. They learned those names of animals in the morning and they had time to stay awake, versus if they learned the names of those animals in an evening session and then they were tested on them later. So if they learned the names of these animals and then went straight into a sleep period, they actually had worse long-term retention than the groups that the group of Down syndrome that learned them in the morning and had time to consolidate that information. So this was the first study to suggest that there were some differences with when individuals with Down syndrome learn information and how much they could retain. And we in our lab have found that there are differences in the amount of language information that individuals with Down syndrome can produce based on sleep profiles. So from two different studies, Breslin et al. In 2014 and Edgen in 2015, we found that verbal IQ was reduced, about a 9-point difference in verbal IQ between children with Down syndrome who were good sleepers versus poor sleepers, based on the level of obstructive sleep apnea they had. So we actually did in-home sleep studies, looked at their levels of apnea, and we saw a difference in their verbal IQ based on that. And then this study was conducted with actigraphy, which is a movement monitoring method, and we found a difference in the number of words reported on the CDI by parents between good sleepers and poor sleepers with Down syndrome. So there had been suggestions that memory consolidation was impacted in this group. We have seen that correlationally, sleep is related to language outcomes. We wanted to then know if we train individuals with Down syndrome on learning new words, so arbitrary object label mappings, could we see that they had differences in their retention over different intervals based on whether sleep was included with that interval, would they be able to benefit from naps in the same way as I discussed before? When they're learning new words, would they show this benefit of that nap like typically developing kids do? And so we conducted this study of nap-dependent learning of new arbitrary labels in a group with Down syndrome that were habitually napping, so 4 or more times per week. There were 25 individuals with Down syndrome and 24 controls. They were matched on nonverbal IQ scores on the leader. They had similar gender distributions, and they were toddlers. So the group with Down syndrome was 53 months, and the typical group was 33 months on average. So they weren't, we didn't have a chronological age match control. We had a mental age match control in this study. And what we did is this was all within subjects. So each of the children with Down syndrome were measured in each of these conditions. They were counterbalanced with different lists. But we trained them on 3 object label mappings, and then we had a 5-minute test where we test the retention just to see, can they hold on to 3 labels after 5 minutes at the same level of typically developing kids. We also had them trained to a criterion level and found that there were no differences on any of the lists between the group with Down syndrome and the typically developing kids and how many times they needed to see these items to retain them. But we basically tested them on their 5-minute retention, and then we compared 2 conditions, one in which we trained them on 3 labels and had a 4-hour period in which included a nap, And we measured polysomnography sleep recordings during this nap period so we could see what the sleep was like. And then we also trained them on another 3 labels and had an equivalent period of wake. So this allowed us to directly compare how much can each of these children retain at 5 minutes, at a 4-hour period, and then a 4-hour period that they nap. Okay. And then we also tested them 24 hours later to see if over another night's sleep they would show similar levels of retention. So from the typically developing literature, we had expected that typically developing kids would show a benefit of that nap. This is what we found. So these are the data from typically developing is in white, and Down syndrome is in black. And at 5 minutes, and this is their training level, as I mentioned, we trained them to criterion, so this is where they were trained to. At 5 minutes, we had equivalent, these were not statistically significant differences here, but equivalent levels of retention in the group with Down syndrome and the typically developing kids. And then what you see here in white are the results after a wake interval in the typically developing kids. So there is some loss across wake in children who are typically developing. And then 24 hours later, it's not improved that much, but they have this loss. But if they stayed awake, they had this loss. But if they actually took a nap, we saw that benefit of the nap. Okay, so they're learning the labels, and they seem to do better if they have that nap at the 4-hour interval, and then they have retained as much as the original training at 24 hours later. So sleep does benefit the typically developing kids in this study, as we had expected. What happens in Down syndrome? Well, what was very interesting, because even though they had this equivalent 5-minute retention, if they stayed awake, they actually did better than the typically developing kids. But if they slept, that's where we really saw their deficit. So they were not retaining as much across these different intervals. We saw typical level of performance at 5 minutes. They were a bit better at 4 hours, but if they slept in that 4-hour interval, that's where we saw the big decrement in performance, and that was also measured at 24 hours later. So it suggests that the timing of sleep is quite important for these individuals, and that they need some time to consolidate, and that there's something about the sleep period and the sleep quality during that period that might be interfering with memory consolidation. So looking at the memory, the EEG findings from individuals with Down syndrome. So the qualitative analysis of the sleep from the ASM coding. So this is a qualitative coding by a coder who's reliable to score sleep studies. And basically what we found was that there was a reduced amount of REM. That was the biggest finding from the sleep studies. They were about the same length. There were many parameters of these naps that were quite similar between the groups with Down syndrome and the typically developing kids in terms of time of day, pulse oximetry, duration, onset delay, the arousals, etc. But the REM was different in the naps. And then there was a subtle difference in the delta range in terms of the power analyses, but that didn't relate to outcome. What related to outcome that we saw was that REM was different in the group with Down syndrome and actually correlated to retention in the typically developing kids. So this suggests that there are memory consolidation differences in Down syndrome in terms of when they learn new information and when they learn that in relation to the sleep period. And that is related to some aspects of that sleep architecture in the nap, particularly it was shown in the typically developing group that REM, having more levels of REM, was better, allowed for better retention. So now I'm going to go on to talk about these studies that we've been pursuing to try to see, given this, this difference we have with sleep-dependent learning and consolidation, whether we can help those with Down syndrome consolidate information through these other memory training techniques that don't rely as much on the hippocampus or sleep. So this is a study that was published in 2018 on fast mapping. And basically in the fast mapping process that we followed from a group that was interested in doing this with hippocampal patients, It involves presenting a known item alongside a new item that you're presenting a label for. So you're basically, there are different categories of items, and you are presenting an animal-like item with another well-known animal to help support the semantic understanding of that and help with integration of that. And in a study in 2011, what they found was that patients that had hippocampal lesions were able to benefit from this technique, that they actually were able to retain information better over a 1-week delay because of having this fast mapping introduction to the items versus the deficits that they had. You can see there the deficits they had with explicit encoding, which is basically when you present items and you're asked to remember them, their arbitrary label with no other supporting information. So in the fast mapping paradigm that we used, and I'm happy to talk about this, and maybe we can talk about this in the seminar later, we had to do a lot of modifications of this for children. We tried different mapping techniques with different methods from the Sharon study and others, but we had to make it very simple, and we turned it into the pointing game where the well-known referent was there, but they weren't explicitly asked to remember the item. So it was implicit, and the referent was there in our procedure. So they were asked to point to the hydrax, which was the new item. And then the explicit encoding, they had 4 items. They were asked to do the remembering game, and they just saw that item and were presented with the label of that new item. And so she had different sessions of fast mapping and coding, so starting with the encoding trial, and then a 5-minute delay, a 1-week delay, and then a familiarity test. And the fast mapping procedure was always presented first, and then she had the explicit encoding procedure. And that basically had 4 items, a 5-minute delay, a 1-week delay. And then they had other familiarity tests and category tests to see if children with Down syndrome could categorize the items at all or were familiar with what they had seen. So fundamentally, they actually all did pretty well. And I'll talk about the various iterations we had to go through to get to this point, because there were some parts of our development of this in which the kids with Down syndrome were at floor, and we went down to 4 items to make sure that they weren't at floor, because in order to look at long-term retention, we needed to make sure they had enough initial performance to do this study. But so they were, that was chance at that line right there. So they were all above chance, but we didn't see any differences amongst the conditions here in these kids. So these kids were typically developing children that were roughly about 4 years of age, and then a group with Down syndrome that was 18 on average. So no difference across the conditions. When we looked at the gain across 1 week, the typically developing kids did have a bit more gain on the explicit encoding. There was no difference on fast mapping. So we didn't see this benefit of fast mapping that we thought we might see based on the previous work from those patients that had hippocampal damage. Looking at the eye tracking, so this was basically whether or not they would focus on the region of interest of that target item during the recognition test. And what we found was at 5 minutes for explicit encoding and fast mapping that there was really no difference in eye tracking in Down syndrome. And for the most part, neither group was showing an eye tracking effect. There's a little here, this is a Down syndrome finding, a little finding late in the trial where that they were actually above chance at that point. But most of all, they're not showing that eye movement towards the target at 5 minutes. We did see this extended effect in both groups, same effect. There was no difference between groups, no interaction, where at 1 week they showed this very late preference to look at the target And that was in fast mapping at 1 week and explicit coding at 1 week, and the same effect emerged. So there was no difference in conditions there. So basically, we didn't see any special benefit of fast mapping after 1 week. We did see learning emerge after 1 week, but we didn't see a fast mapping benefit. And we also saw no relationship with sleep. So looking at explicit encoding versus fast mapping and the correlation between sleep, there was none in either group after 1 week. This is just presenting the Down syndrome results because that's what group we're most interested in at that point, but no correlations between sleep efficiency and change across either of these conditions. So fast mapping did not reveal the results that we had hoped in terms of giving a benefit to Down syndrome for those mappings. So moving on to self-reference, this is in collaboration with my colleague Matt Grilly at the University of Arizona. There's a memory advantage for information that's encoded in reference to oneself. So if you're processing a new concept or learning a new thing and thinking about how you would interact with that thing or how it relates to your life, you tend to remember that better. We know from studies of aging that that's one of the ways that older adults seem to retain more, that if they are processing new words or concepts in relation to things that are relevant to themselves. They also show a benefit in hippocampal amnesia and patients that have memory disorders from this technique, or patients with diffuse injury. There have not been a lot of studies looking at long-term delays, and we decided to do that in this particular study. And we wanted to know, could encoding with self-reference help individuals with Down syndrome? So there have been studies suggesting that concept of self is something that's reliable in individuals with Down syndrome, that they have developed a self-concept. So we wanted to know whether or not we could use that to help support memory retention in this group. So this was 26 individuals with Down syndrome, 10 to 27, 25 typically developing kids. And there are lots of different tasks that we administered as controls. So we did a self-concept task. We assessed whether or not they knew the idea of ownership, because our paradigm was about ownership. It's basically asking them how excited they would be if they got these toys, how much they would like a particular toy they were assigned. And we use this particular self-reference measure because in the previous literature there had been suggestions that younger kids could remember more if they were, processing objects in relation to liking and owning that object. And so we had them encode different items, different toys in relation to themselves. And then we had a 1 week delay that included actigraphy and sleep diary, and then we had a recognition of those items as well as other sort of control measures that I can talk about if you're interested in. So the task looked like this. They had encoding, so we had 2 researchers in the room and the participant, and they were presented with a toy that had a border of a certain color, and they had to sort it into these boxes of that color, okay? And each of the boxes belonged to either researcher 1, researcher 2, actually it belonged to researcher 1, the participant, or there was no one who owned it. So the researcher that was on this side of the table was just directing the experiment. And so it was basically sorting cards and giving them to each other to determine whether or not, the question would then be whether or not they liked it a lot, a little, or not at all. If the participant got the item, they would have to make an assessment about whether the researcher liked it a lot, a little, or not at all. And we had some introduction to the researcher so they could answer that question. And then when the box that no one owned, that we asked them to judge the size of the items, just so they were doing some encoding of that item. So it was really just sorting of these different items and asking how much they would be liked by the person themselves, which was the self-reference condition, versus another researcher. So that was how the items would go into these different boxes based on that. And then in the recognition test, they saw that same item, but the border was now removed, and they would have to put it into the box it was originally sorted into. So it was either the box that they had the color they had, the color the other researcher had, or no one had. Okay, so assessing whether or not they could sort them into these boxes and actually remember the items that way. Like that. So I'll start with telling you the results of just the self versus other condition. Okay, so overall, there was no self versus other effect in this particular study. And it's possible that the way that we introduced these items was all sort of massed together. And so the subject would get one of these items and would sort it by the self, and then one sort by the other. And we might have had a better benefit of self if they had processed all of them together in reference to the self and then moved on to processing the other. But in particular, we didn't find any benefit of self with the way that we administered these tasks. What we did find is a benefit in typically developing individuals in a group by condition interaction in which the typically developing individuals seem to benefit more from the size estimation of these objects. So actually encoding it in relation to the object size seemed to support the typically developing kids more than the individuals with Down syndrome. We found an overall group effect where kids with Down syndrome had memory deficits, and then the size seemed to benefit the typically developing kids better. So basically another study that we have run where we were not able to find that self was beneficial to Down syndrome. So now we have fast mapping not working the way we thought. We have self-reference not working the way we thought. And sleep in this particular study was not related. So both of these studies, the fast mapping study and this study, seem to not show much relation between these different memory processes and sleep. Now I should mention that in both of these studies, we made them very, very simple so that they would have the best chance at retention and long-term retention as we could get. And this, one of the things we're thinking is that the sleep had not related to some of these outcomes, particularly in the fast mapping study, because retention was high, and we didn't give them a big enough memory load to show those relationships. Okay, so moving on now to retrieval practice. So this is the idea that we are better at retaining long-term if we are tested on information versus just seeing a repetition of that information. So if you're studying day-to-day for your classes and you just sort of read over a textbook, that's not as good as if you quiz yourself on the answers to that textbook. And so in some previous work we had done, both with Bruce Pennington and work in my lab, we'd seen that on list learning type tasks, tasks like there are word lists in which they have to repeat multiple words. The experiment repeats multiple words, and the individual with Down syndrome has to learn this list and repeat them back to the experimenter. We found that they were worst on the initial trials of those tasks. So that looks like on trials 1 or 2 or 3, they have the biggest deficit there. But after those repetitions, they actually seem to catch up. And we've seen this in these particular 2 studies, but it's something that's reoccurring. So it seems that they can benefit from repetition. Now, on a list learning task, they're both getting the exposure, they're getting a repetition through the experimenter repeating that list, but they also have to generate that. So this particular task kind of muddles the testing effect in that there is repetition as well as retrieval. So we wanted to design a new task that would allow us to look at repetitions versus retrievals and also be able to look at long-term retention and whether or not we could basically compare side by side what would happen when someone was given a repetition of information versus a retrieval, and then with the control condition of just time spent processing that information. And we used deferred imitation, which is basically a very standard memory test that is used by Patricia Bauer and many individuals for years, in which children are shown arbitrary sequences of events. So we have 3-step sequences, and I'll show you an example of them in the next slide. And so basically have these baseline interactions with the items to see whether or not they'll generate the actions on their own. And we piloted all of these to have the least baseline generation that we could. And they're asked what you can do with these things, and then we look at what they can do with them. But then they're modeled twice by the experimenter in terms of the full sequence of 3 events. So that's the model test condition, in which they're modeled twice, and then the child is actually given the items, and then the child is asked to demonstrate the sequence, which is basically a retrieval of that sequence. Okay. They were either modeled 3 times by the experimenter, or they were modeled, and then given a pause of 20 seconds, which was basically to allow for processing time versus a test versus another model. So we're basically comparing a test opportunity to a another model of the sequence versus time to process. Okay, so we wanted to know whether or not this was going to be the best condition for individuals with Down syndrome compared to the others, based on retrieval practice benefiting in our previous data, but also that there's a wide body of evidence to suggest that retrieval practice seems to be more beneficial and actually taps different neural systems that could be utilized by this group. So 1/2 of these sequences were tested at 5 minutes, and then there was a 1-month delay in which we tested all of the sequences. And then the second time that they visited, we had these interactions with the sequences, and the children were asked to show what they had just done. So this study was 30, and this is ongoing data collection, so we're hoping to collect up to 90 individuals with Down syndrome in our multi-site project with this particular task and other tasks that we're validating for memory assessments. But this was what this tasks look like, just to give you an idea. So she's now modeling that. And then she pushes it to the child to allow for a retrieval. Okay, so there were 6 different sequences like that. So at 5 minutes, and remember what we're comparing is whether or not they had a test opportunity, another modeling of the event, or a pause, right? So interestingly, at 5 minutes, the typically developing is in orange, Down syndrome in blue. There was an effect where typically developing children looked like they had better 5-minute retention if they had a pause opportunity. So if they didn't get another model or test opportunity, they had better 5-minute retention. There were no other condition differences across the groups for that. So looking at 1 month, whether or not the sequence was tested versus not tested, it benefited both groups to have a testing at 5 minutes. So to have another opportunity at that 5-minute delay to have another testing of those items. So this is looking at those sequences that were administered again at 5 minutes versus those that weren't. And both typically developing kids and kids with Down syndrome had a benefit of that testing. So this is important to keep in mind, because a lot of times when we do memory tasks, we do repeated retrievals, especially with young kids and with kids with Down syndrome, and that's because we want to solidify the memories or make sure that they're actually getting the information that we're presenting to them, and it can have very different outcomes for long-term delays to have those testing opportunities. So looking at 1 month with retrieval practice, what we do see is that the Down syndrome group looks like they have better retention in the testing condition here. So we have pretty poor retention of the pairs of items. As you can see, those are associated pairs across both groups. But we do see this difference here between testing and modeling, and we also get it with actions. There were less strong effects in the typically developing group at 1 month. But what we do see is we get these differences here with the modeling being better for typically developing kids. And what we see is that sleep is beneficial in this particular task, but only for the pause condition, so the one that basically had less demonstrations. So there is a finding in the sleep literature that sleep tends to benefit more weakly encoded or items that have fewer retention, and so fewer repetitions. So that might be one of the reasons that we found a sleep effect in this particular task and for that particular condition. So that's only in Down syndrome we found that the pause condition was influenced by sleep. So just to sum this up, does sleep matter? In looking at the sleep effects across these different memory interventions, we found no correlation in fast mapping with sleep, no correlation with the self-reference effects in sleep, but retrieval practice, we did find a correlation in Down syndrome with sleep quality correlated with better retention in that pause condition only, and it may be because of a weaker encoding of that information. So if information is really strongly encoded and people are at ceiling prior to a long-term delay, sleep may not be of benefit. So just an overall summary, memory deficits are not absolute in Down syndrome. I hope by seeing these different intervals that we presented information and different outcomes across retrieval practice, across different intervals that include sleep, they have impairments in relation to chronological controls. They would have impairments across a number of different areas, but in relation to mental age match controls, we can see that there's some areas in which they have more preserved ability, and that can be based on the timing of when they learn information in relation to sleep. It might also be in relation to encoding presentation as well, that we can get better retention based on how it's encoded and when it's presented in relation to sleep. The presence of napping or sleeping right after learning may be particularly problematic in Down syndrome. We don't see that rapid or implicit methods of cortical integration like fast mapping or self-reference, which we think is a rapidly occurring event, those don't seem to work in this population. Understanding why, I think, is an important thing to do. But with the repetition with testing and having additional testing opportunities does seem to impact long-term retention in this group. Sleep tends to impact weakly encoded memories more than strong ones. That's something from the typical sleep literature, something that may be reflected in our retrieval practice findings as well. And that might be one reason that it's very important for language. We don't get language is rapid, it's arbitrary often, and it can be very, for young children, getting all of that information coming at them at once without repetition or opportunities to practice. That might mean that they need that sleep interval and that those memories are fragile until they get them. And then in general, to understand how to best support long-term retention in all the groups have intellectual disability, including Down syndrome, we need to know how best to present information and how those memories may interact with sleep and sleep deficits. And I think one of the things that these findings also emphasize is that we can't just look at a 5-minute interval and say that those memory findings are going to translate into what will happen across the long term. So in our study of retrieval practice, typical kids look like they had better memory with a pause. We wouldn't want to go and intervene in that way because that didn't last across a month. But knowing what might last over the long term, I think, is quite important. So I want to thank the Trisomy Research Society for funding this, Natalia for inviting me and putting on this wonderful conference, and then my lab and all of the people with Down syndrome we work with, and all of the funding agencies that support this work. So thank you very much for your time and attention. Appreciate it.
¿Alguien tiene alguna pregunta?
Wonderful work. Thank you so much. I am wondering if you have ever studied the procedural memory and the sleep?
Procedural memory?
Right.
I've not done that with Down syndrome. No, I focused more on explicit memory. I think it might be important to do that. Yeah, I haven't. I think there has been a study that has looked at. Natalia, do you know of a sleep-dependent learning study that's looked with procedural memory in Down syndrome? There's a lot of work about, from, there's a group that's shown that kids actually sort of go from implicit to explicit memories with procedural kinds of tasks. So I think it would be something good to look at. Is there a particular reason that you were interested in that area?
Yeah, because that kind of memory is non-hippocampal dependent, right?
Yeah,
it's in
yeah.
different regions.
Yeah.
So I was wondering whether the mechanism of the sleep, especially the REM sleep, is also affecting these other types of memory.
Yeah. I think that I've stayed away from that because it's gone off and on about consolidation on those implicit tasks and the role of the hippocampus in that. There's some camps that believe that that's non-hippocampal completely, and there are others that believe that the hippocampus is functioning on some of those more procedural level tasks too. So that's one of the reasons it hasn't as excited me as much, because I felt like the primary story and data about sleep-dependent consolidation has related to arbitrary bindings and explicit tasks. But it could be, now that we have those data, it could be really interesting to start to contrast, okay, does Down syndrome look like it does have some types of sleep-dependent consolidation that are intact, right? So that could be a very interesting question now that we've gotten to this point. Yeah, thank you.
How do you explain that children with Down syndrome have a relationship between the sleep and vocabulary, but there are no relationship between the sleep and mechanisms for word learning?
I'm not sure I understand that. So which part, so are you talking about the final correlations?
Yeah, I work with good sleeper and poor sleeper. Good sleeper have better vocabulary.
Yeah.
But in your fast mapping study, you don't have a relationship between these two variables.
Right. So there is one study that I presented in which we do show when we're training them, right? When we're training them and we show the sleep intervening, that consolidation deficit is there. But and we saw differences with actigraphy in that particular study with the younger children, right? So my only explanation for that is that that we overtrained them. That so basically that we actually, in that fast mapping study, we made it so easy, and we also tested them again. So we gave them a few number of items, because when we gave them 8 items, they were at floor. So we had to get the items down to 4 to basically get retention that was above chance. And then we tested them again at 5 minutes on the entire set. So I think we gave them too much support, which is great to give that much support, and that's what you need to do to make sure you don't have really low levels of learning initially. But I think we gave them so much support that it's not something that required sleep. That's my only explanation for that. And it's a really important question. I think that's really key to understand. Under which instances is sleep important in Down syndrome? And can we understand that? And can we understand the ways that they don't require sleep to consolidate something? because that's the way that they'll be able to learn best, I think. But I think I also think that, there are more data that need to be collected with long-term delays and with sleep as part of that picture to understand these correlations. I mean, we've started to do this, but I want to see it done in a lot more studies because, getting 20 kids and understanding what's going on, whether or not this is a real lack of a correlation or it's a power effect, or, I think it's good to just keep asking that question and keep trying to understand what's going on with that. Yeah, so that's my only explanation for that. It's an important question. Yeah.
Thank you.
Yep.
Well, I have another question. Do you know worse than study the relationship between lexical networks and sleep?
Yeah, the work that's looking at the lexical relationship, I think that that's a really cool thing that could be done. So semantic network mapping, I don't know that anybody who has done that, but I think going in and taking, you could go in and take CDIs and do that and look at sort of semantic networks. Is that what you mean? That would be, I would imagine that they would have less developed semantic networks. And there, Plunkett's group, other people have been doing that. I don't think they're doing it in Down syndrome, but you could do that kind of thing. I asked a student to do something like that a year ago, and they're like, no, it's too much work. But we have, we have Lena monitors that have language data. Len Abaduda's lab's been collecting language samples and narratives and all. I think you could there are lots of corpuses of data where you could do that kind of thing and look at that. And I would love to talk to you about that more. I think that's a great idea. I don't know if anybody who's actually has anybody just done that in Down syndrome? That's cool, right? And then to look at it in relation to sleep would be amazing. That's a really terrific idea. You should do it.
Thank you.
Yeah.
¿Alguna otra pregunta?
Thank you for your talk. You presented data where they have these colors around the cards, and they have to put the cards in the experimenter or the. Okay. So I was wondering, there's this research in teaching where they put together two sensory inputs. So, for example, to learn molecules, they put the molecules, so the visual input, with the auditory input, and apparently it's better if you have the two. So I was wondering that instead of having two inputs that are visual, because they may be concentrated in the color, you had an experiment where there's the card, but also there's the input. Because actually they have to remember location, color, and card. So I was just wondering, what's your input on that?
I think that's that's a really good point. I think there wasn't enough processing time in general. I think kind of showing the item and then saying, this is yours, or, something. We used this paradigm because it was a child development paper that had shown an effect in typically developing kids, and we were just starting there, hoping that we would get the same effect. But as she started to modify it, we said, well, we needed another encoding condition, and we blocked everything together so that, they got the one card, and they got the next card that was the other, and the next card that was, the didn't belong to anyone. And I think that it probably would have been better to have it more processing of the self included, and potentially have it so that there were multiple inputs. Because I absolutely agree with you. I think for kids with Down syndrome, having all of that, it's a simple border, but avoiding that kind of situation as much as possible where they have to process something on two concurrent visual inputs, or too much language at once, is really important. So for just going back to the fast mapping study, We piloted that fast mapping procedure with the original paradigm, which was to ask them to point to the one that had the tail pointing up. And when we did that fast mapping procedure, which basically referenced visual perceptual details of the unknown item, we could not get them to map. So they were not able to pick the unknown item in relation to an elephant when we had that much language. So we had to go very, very simple. So I think you're right. I think that there are multiple ways that we could have done better with that particular paradigm. It's a trade-off, though, kind of doing something that showed an effect earlier versus, you know, yeah. But we didn't see that initially. But it's a very important point, yeah.
Another question is, so the time of taking a nap or the time learning. So I think that for example, the work Blanket with typical kids, they probably haven't taken into account those times. So the child can either come to the lab in the morning, so learning in the morning, or learning in the afternoon. So it's very, I don't have a question, it's just very interesting that we have to take into account other variables. When is this learning occurring? When is this napping occurring? Or the sequence of sleep and wake, when is this sequence occurring? So
yeah. Yeah, it is really interesting, and I think a lot of times when we, neuropsychologists and memory people in particular, we don't think about intervening naps or, when information is presented in the lab, or getting kids into the lab at a similar time of day to when they were first tested. And I think it's something that is really an issue for these developmental disorder populations, that they need to, we need to take into account whether or not that's going to happen, and think more about it in our design. So, yeah.
¿Alguna otra pregunta?
Well, I got a little lost, and maybe because I'm dumb. But
no, I just talked too fast and wasn't clear.
I quite didn't really understand why on the task about the boxes you had a box and no one box. Right?
The unknown box? Yeah, I'll go back to that.
No, but you were testing size and not the likeliness. And
yes.
I didn't understand why.
Yes, I'll explain that. So basically, what we were trying to understand was when they're presented with these toys and they were given them in their box, and then they rated, how much they rated them how much they like them for us to control for, like, the best toy that they've ever received versus like the thing that they didn't care about, right? So we had to give that to them, that rating, to help us understand how much they like that thing or would be happy about getting it in their box. And then we were comparing that assignment to that toy to them versus when it was given to someone else in terms of how much they would remember. But we had to have another option to see whether or not they could remember who it was assigned to in the first place. And so that's why we had the other box. And in order to have them process the other thing in a similar way to the self versus other, or the box that wasn't assigned to anyone, we need to give them a rating of something. So we used the big versus medium versus small rating for that particular assignment. So the green box was no one's box, and it helped us to understand whether or not they even remembered the objects at all. Okay, and then that was just a way of equating ratings of the items.
So you wouldn't expect to be a difference if there's a no one box and the children have to rate how much they like the toy?
We didn't want them to process it in reference to themselves for the no one box, right? So we asked them to process it in a different way, which turned out to be in typical kids the way that worked best. But, you know, it was a way of giving them a processing task. Because if they just stuck it in the other box, we wouldn't know whether or not it was that, you know, we wouldn't have that same kind of rating on it. Yeah. So. Yep.
If they are remember things from selfness, wouldn't it be like bias or a help of you to have multimodal representations for objects, not only visual cues?
Yeah.
Because they have like more proprioceptive information about the thing.
Yeah, you know, and a lot of times when this work is done, with kids it would have been good to have all these things, but you know, there's a lot of things that we showed them. But in older adults, when this work is done, they're asked to imagine themselves interacting with the item, right? So the item's presented, and you know, it's a group of things like a coffee cup on the ocean, and you have to think about yourself walking on the ocean with your coffee cup, right? So that's a way of imagining your body interacting with those things. It's not, you know, proprioceptive in a sense, but it is like an imagining event, and so it is more of an integrative event that like you're talking about. And I think you're right, like if we had done something of where they could hold it, you know, with less items, that we may have had a different, you know, like what is it like for you to have this thing now, and what does it feel like to you in your body? Like those are all things that are really important when you have that self understanding helping to support the memory. Yeah. So good question. Thank you. No, that's okay.
I also like a little your eye tracking task.
Mm-hmm.
I was wondering if you could explain a little further about the temporal windows.
Yeah. I have to remember now. So I think it's not easy to see them. Sorry, this didn't project very well. But so we went 0 to 250 windows up to 2 seconds. And we use that interval because a lot of memory eye tracking tasks in children tend to go up to about 2 seconds, and see sort of, you can see later memory effects in those later windows. But you know, here in the 5 minute, so it's basically tracking their level of looking time to the particular target, and they were presented the target, one they didn't see, and then one that wasn't that label, right? So one that was labeled but not that particular label, and then a distractor item. And then so we basically didn't see above chance looking anywhere except this window here for Down syndrome and this window, which is later. I think it's. I'm sorry these didn't come out. It was really bad slide making on my part here, but yeah, I can send you this paper if you're interested. Yeah, it's published. So, and I think in the paper we actually can see the data. I'm sure that that's the case, but it looks like 250, 250, 500, 500, 750, 1,000 at that like 1,000 to 1,250. We see that effect. But then it was really no different overall. No interaction with the typically developing kids, and then this later window about at that 1,200 and later is when we saw the viewing of the target 1 week later. So it's interesting because it's stronger at 1 week later. It's a stronger effect with the eye tracking than it was at 5 minutes. So, yeah.
It what happens in the post-naming window?
It's while they're being, so basically what they had to do was identify which one they had seen, and they pointed to it. And it was prior to that they were given the presentation, and it was concurrent with the label. and we were looking at what they were looking at during the label presentation. So the auditory stimulus came up, they had the 2-second window, and then they pointed. And we did a lot of that because where we had windows of time where we were just eye tracking, because I was worried a lot about like the pointing event interfering with eye tracking. So we would freeze it for 2 seconds and put up the stimuli so we could see where the eye would go when the auditory stimulus was delivered. Yeah. Does that help? Yeah, okay, great. It's funny, I can see it on here, but I can't see it on there. Yeah, should be looking on here.
¿Alguien tiene alguna pregunta en español y aquí ayudamos a traducirla? O también en redes o en YouTube, no sé si, ¿no?
Sorry, I was just thank you. I was kind of interested right now at the behavioral data that you got from the pointing game. That was kind of interesting as well. So, what can you tell us more about this pointing game they had, and also their performance about 5 minutes?
Yeah. So. Basically, in particular, we didn't really see very much differences across these intervals in the different different procedures. The 5-minute interval was a complete test of the item set, though. So they were presented with the items, and then they were asked to point to those items. And I think she also did. I have to go back to the paper, but I think she also did 3 presentations of the items at that point in time. So at the 5-minute interval, I'm pretty sure she tested them 3 times. And we did that because we've learned now not to do that. But we did that because we were worried that we might have like sort of random. And we had a small set of items. So we're worried about sort of random or non-attention to those pointing. So that was, there was a lot, it was more testing than I would do now. And it may be in part why we have sort of higher levels of retention at 5 minutes than I would expect. Because, you know, they are arbitrary labels, and I would expect there would be some loss across 5 minutes generally, but we didn't really see any significant loss at 5 minutes. And then having no loss at 1 week is really unusual, I think. For most of our studies, we do see loss across. So the self-reference study, we saw loss in both groups. We had the Down syndrome difference. And most of the studies we run, the sleep-dependent learning NAP one, we didn't see loss at 5 minutes in Down syndrome. But most of the studies we run at 1 week were definitely showing loss of some sort, both in typical kids and kids with Down syndrome. So I think it's unusual that we don't have any reduction in performance of this over 1 week, and that we have this eye tracking thing emerging. So my only explanation for that is that we overtrained them, and that we made it really easy. So, yeah.
Do you think that this would be correlated with another kind of, behavioral test, or that kind of assess memory, this behavioral pointing? I don't know. Do you think that would be possible or not?
Well, I would have expected that the explicit encoding, if it was difficult enough, would be correlated with many kinds of hippocampal. I mean, that's from the literature. The mapping of a single item onto a label is a task that we think is an arbitrary binding. So I would expect it would be correlated with a lot of hippocampal-like tasks. I can't remember if we ran anything like that. I don't remember running anything like that with other tasks. But yeah, I mean, it's possible that if we ran those kinds of, like this paradigm versus another one where we didn't test them as much, we might see more correlations of that sort. Yeah. We did wonder whether to publish. I think this is an interesting story for students. I'll just quickly say we wondered whether to publish this, but then we thought we did this. We didn't, and we thought like the retention was interesting, that we got the retention, and she did it in a really controlled way. And during the editorial process, they had actually told us to take the sleep findings out because they weren't expected. And I said, I don't think that's a good idea. Like, we did it, it's there, it's not correlated. Let's figure this out and see if it helps, people to understand when sleep is related, when it's not. And so we're happy we did that in the end, because I think it's important to be clear. And, if you, so you don't always have to have a positive finding, right? And I think it's important to understand why in some ways and, why not. Yeah. Thank you. Thank you. So sweet. Thank you so much.
Bueno, pues desafortunadamente ya acabamos. Muchas gracias por acompañarnos y muchísimas gracias. Thank you so much.
Yeah.
Y al maestro, casi doctor, y al doctor Mario, muchas gracias por acompañarnos y aprendimos muchísimo. Natalia al principio dijo que ojalá aprendiéramos, pero nos quedamos con ganas de mucho más. Y gracias a todos.